Before Dr. Stavitsky describes a little bit about Enrique Ecker and introduces our speaker, I just want everybody to know that this is the second lecture in a year-long series of lectures commemorating both Darwin and evolution as a subject. And we'll be having a number of other speakers in all different departments and throughout the university up and through April of 2009 in connection with this celebration. So without further delay, Dr. Stavitsky can take over.
This series was initiated in 1957 when Professor Ecker retired after 40 years on the faculty of the Department of Pathology. Ecker was a pioneer in both bacteriology and immunology. However, his laboratory, he and his students and students were pioneers in the study of the complement system. They were pioneers in working out the sequence of activation of the complement components. Ecker's student Pillamer conceived the alternative complement pathway, which I think is one of the most brilliant hypotheses of the last century and has turned out to be a key pathway in both the cause and prevention of disease. Pillamer's student Erwin Liebhau and his students discovered the enzymatic nature of the complement system, which promoted the further study of the pathway. We're very fortunate that elegant studies of this pathway continue in Cleveland, in Laura Nagy's laboratory at the Lerner, and here in the laboratories of Ed Medoff and Christopher King. It is fitting that as part of the Darwin celebration, this year's Ecker lecture deals with the subject of evolution and immunity because the complement pathway is one of the earliest pathways to develop, and Ecker and his students focused on the study of the relationship between infections and complement. Gary Littman, this year's Ecker lecturer, received his PhD from the Department of Microbiology at the University of Minnesota. Incidentally, that's the department where I received my degree a few years earlier. A brief summary cannot really convey the diversity and richness of Dr. Littman's contributions, which were made in several quite different venues at the University of Minnesota, Sloan Kettering, and for the last 20 years or so at the University of South Florida in St. Petersburg. At the University of Minnesota, his mentor was Robert Good, who I always felt should have gotten the Nobel Prize for his linkage between the thymus and cellular immunity. And there, they studied the immunoglobulins and they did landmark studies of the immune response in the lamprey, bowfin, horned shark, turtle, and lungfish. I vividly recall this era as a very exciting era in unraveling the phylogeny and ontogeny of the immune response. I recommend that you look at these early papers from this group, as well as the remarkable series of papers by Arthur Silverstein on the ontogeny of the immune response in the sheep fetus. At the University of Minnesota, he was director of the Clinical Laboratory of Immunochemistry and an assistant professor in the Department of Pathology. He then moved on to Sloan Kettering as an associate professor in genetics and later immunology and director of their immunochemistry laboratory. He became chairman of the biology unit of the Cornell University Graduate School of Medical Sciences. At Sloan Kettering, he continued his studies of immunoglobulins from different animals, but also worked on the structure of cryoglobulins and erythrocyte membranes. He then moved to the Tampa Bay area, and from 1985 to 1990, he was chairman of medical genetics at the Tampa Bay Research Institute. In 1986, he became professor of medical microbiology at the University of South Florida, also Heinz Professor in Pediatrics, Professor of Biochemistry, and Director of the Laboratory of Medical Genetics. In 2004, he became Distinguished University Professor, and in 2005, Director of the Children's Research Institute. He has continued his studies of immunoglobulin evolution. He studies T-cell receptor gene homologs in primitive jawed vertebrates, and more recently, immunity in zebrafish. He was part of the large team that unraveled the genome of the sea urchin, which I found remarkably has over 100 genes for toll-like receptors, many genes for the complement pathway, and many genes for the coagulation pathway. He's written a number of theoretical papers on innate and adaptive immunity and the evolution of immunity. The title of today's talk is Diverse Immune Diversity, What Would Darwin Think? Please join me in welcoming Dr. Littman.
Thank you, Abe. I didn't know I'd done all those things. It's been a long time. This is a non-hurricane scene from St. Petersburg, Florida, of the Children's Research Institute. I told everybody I'm very happy to have made it here. It was a little sketchy on Friday afternoon when the tracking models, which I probably know more about than I know about the topic I'm going to talk about today, I had the hurricanes aiming themselves right at this building and the immediate vicinity, but as I think those of you know, it took a turn. It's just a great honor for me to be here today. I'm not a great Darwin scholar by any means. I have, however, as Abe indicated, had a very long-standing interest in the evolutionary processes that govern the diversification of the immune system. And what's going to come out of this, I hope, for you, and I realize that many of you here come from a very medical background, is an appreciation for number one, the enormous diversity that exists, number two, some very important alternatives that have evolved, and most significantly, how dramatic changes have been over, in Darwinian terms, a relatively short period of evolution. This image is of the New York Botanical Gardens recreation of Darwin's gardens in Kent, England at Down House. And it certainly shows me something that I already knew, which is that Darwin was a great observationalist. He had just phenomenal powers of observation. He could examine, and I have seen some of the correspondence, minuscule aspects of the taxonomy of an insect, and at the same time ponder why a long extinct species skeleton was found in a mountainous region. It's really amazing to read some of the firsthand accounts of his work. But something that likely would have puzzled Darwin to quite some degree is adaptive immunity. Now, I also know today I was told that a lot of you are not immunologists, and immunology can be a overwhelmingly complex subject. So if you don't mind, for those of you who are very, very accomplished immunologists, if you would just bear with me for a minute or two, I'd like to get everybody up to some basic understanding of some of the terminology so that the work on the evolutionary diversification will become a little more clear. And we might as well start out with the most perplexing of the issues, and that is adaptive immunity. Now, adaptive immunity is, for what most of you know, is what we consider to be our antibody response or our T-cell receptor response. We're challenged with something, our body reacts with remarkably specific and phenomenally complex receptors. And our main model, as you can already see from the image that I pulled up, is that of a mouse. Mouse is the evolutionary model that we use for looking at human. It's certainly a highly adaptable laboratory animal model, and it has been a powerful, powerful system. The evolutionary distance between man and mouse is relatively long, and there is really very little about modern immunology that has not come from, or at least been confirmed in this animal model. Now, the molecule that's used as a primary reference in most of this work, in most of our immunologic thinking, is an antibody molecule. And here we have a ribbon molecule of a model of an antibody molecule, and it has a polypeptide chain in red and another polypeptide chain in red, and they're really the same sequence. It's just a dimeric molecule. And we have a covalent bonding of another polypeptide chain that's shorter. This is a light chain, that is a heavy chain. and they interact at their ends, if you will, with foreign material of all kinds and character, with remarkable specificity that is formed in a very unique pocket. And the challenge that all of us have in this regard is how exactly, and certainly prior to 1972, what most of us had is, how can you possibly encode in your genome enough information to possibly make 10 to the 14th different molecules? Even if these were 1,000 nucleotides and the genome were compacted, you just do the math. You'd run out of room very, very quickly, And the core of that mechanism from just a standard textbook is that an antibody response really derives from a single cell, effector cell, and that cell has undergone a genetic change in it. It's undergone a very unusual form of somatic recombination. The somatic recombination basically selects, in a very much DNA-driven manner, a single variable region, a single shorter peptide region called a diversity segment, and another segment called a joining segment. And it brings these together in the genome, and by the way, the space this occupies is under 1 million base pairs. It brings together one each of these, a V, a D, and a J in a two-step process. And in doing so, it basically brings together segmental elements that have different sequences. However, anyone would quickly realize that if you have, say in man, 60 or a few more functional variable regions and another 20 or 30 short diversity segments and another 6 J segments, that really is not a very large number. The large numbers in all of this come from an effect when the V, the D, and the J actually fuse to one another. There is the generation of considerable diversity. And that is most easily shown in this extremely simplistic but fairly accurate image. A variable, any diversity, and a joining segment has in effect an enzymatic reaction that takes place at the 3' of the V, at the 5' of the D, the 3' of the D, and the 5' of the J region. And I've drawn a little ruler of sorts. And in a completely untemplated manner, there is the enzymatic introduction of new nucleotide information so that ultimately we create a molecule that bears some resemblance to what originally rearranged, our black, our gold color, and our J, but that there are 2 new patches of nucleotide sequence introduced. And of course, errors are made, and it goes out of frame, and those are eliminated. Autoantibodies, things that are reactive with U, are formed, etc. But at the end of the day, it is this generation of diversity that creates most of the massive repertoire that exists in man. So you have encoded one thing, your single somatic cell has created another molecule on its surface. And again, in a very, very simplistic sense, and really the last of, or next to the last of the introductory slides, we have a situation where, having created that molecule and having placed it on the surface of all of these different lymphocytes, all of these molecules, for all intents and purposes, have a different structure on their surface, that there can be an expansion that is driven by stimulation. That expansion can have several cells in it. The very nature of the locus makes it very prone to somatic mutation at between 10 to the 3rd and 10 to the 4th higher rate than all other gene loci. And that somatic mutant having a somewhat higher affinity, if you will, in simplistic terms, can now trigger the next expansion where it is the favorite expanding member, and another, and another, and another. So almost in a Darwinian sense, we have antigen here driving somatic expansion of a cell lineage. It's not changing our organism. What it is changing at this time is really one individual cell lineage. So our typical considerations of the effects from the outside world of influencing the selection, the process of natural selection, are rarely different. This is a clonal selection. You are not inheriting this molecule at the end of the day. What you're inheriting is an entire system, and it's that system that we're going to talk about in terms of natural selection. So one of the early features of, or early contributions to this was that certainly this process, basically, as I've shown you, has a very long evolutionary history dating back to the cartilaginous fishes. So you name it, cartilaginous fish all the way through man and mouse basically uses a process of diversification that puts a receptor on the surface of an effector cell molecule. So if you go to Darwin in the classic sense and see, apparently Darwin was a very, very poor artist, and I didn't know that, and he really didn't spend a lot of time on his artwork, but he was a great thinker, obviously. And this, of course, is one of these tree of life concepts where forms give rise through common ancestors to other forms. This was truly, truly revolutionary work in its day. We really couldn't do, despite the brilliance of it, it really doesn't pertain to the last example that I gave you, because number one, the system itself is not really acting in quite this way, and I'm going to show you what we do know about it at this time. So I'm going to show you this image twice, and I really, if you possibly can, just focus on the more or less the right-hand side of it. And so we had Darwin's tree and things giving rise to other things, but what is so very, very striking, and it's very out of the norm from considerations like a finch beak or many of these other phenotypic traits that Darwin examined, are the massive differences that exist in the system itself. So we had man and mouse, and they sort of generate diversity the same way, although the immunologist here would quickly tell me, well, not quite. But certainly not in the manner in which an avian generates diversity, in which both in what I should now describe as the B cell system, which is where our antibodies come from, and in the equally rearranging and probably more complex T cell receptor system, which affects cellular immune responses, we have a process that involves gene conversion. There is a joining event that's been preserved, very Darwinian in nature. V and J join to each other in the case of a light chain. However, it's patching of large numbers of strips of genetic information over that variable region, not really the end region diversity, not really somatic hypermutation that's driving the system. And then when we switch ourselves to this system, which I already said, well, that looks just like the system. And I said, remember, the clonal selection end of it is conserved from man through to a cartilaginous fish. Animals such as shark have literally hundreds, not one gene locus, as I showed you in that instructional slide. They have several hundred loci, each consisting of a V, they use 2 D genes, and a J region. Yes, they join. Yes, they create diversity, a very conserved element, but the organization of the system is completely different, and then about half of these loci don't even rearrange. They're already rearranged in the germline, and they are selected. So we have a system where there's selection taking place in the antibody system, both at the somatic cell level, which is the unique generation diversity, and then you're inheriting already rearranged genes. Now, we can't do that. If we arranged our heavy chain locus, we'd have one specificity. They accomplish this feat by simply rearranging by basically joining half of several hundred genes. It's really no loss. It just takes up genome space. Now, the logical progression of thinking in the I think mode would be, well, then the jawless vertebrate must have some remnant of this, so one would think, and the answer is absolutely not. Jawless vertebrates, which are certainly highly derived, there are only two extant forms of them, jawless vertebrates don't use anything like this to generate an adaptive response. Zev Panzer and Max Cooper showed a number of years ago that it's really the modification of leucine-rich repeats through a recombination process depicted in this diagram where different leucine-rich repeats are brought together in a molecule called a VLR. So we can't apply any type of standard logic to this end of immune diversity. This is a completely unique system, whereas this one, be it copy choice and gene conversion, which are the competing models for it, whereas all the vertebrates do at least share a common means of generating antibody diversity, but they manage to do it in functionally different manners. Now, where perhaps it's easier to understand things is innate immunity. You probably gathered fairly quickly, and the immunologists here already know, that it must take some time to make an antibody molecule or a T cell receptor and to select it, get it up on the cell, expand the cell population to enough individual cells where it's really going to have a physiologic impact. What about the rest of immunity? Is there anything else to it? And yes, there's an equally, if not more important arm of immunity that simplistically I'm defining as innate immunity. It's already there, it's on the cell surfaces, it's inside the cells. It's our immune system that does not require elaborate genetic rearrangements and all of its selection taking place at the somatic level. And it didn't take us too long to consider something, and this goes back a number of years, and as I'm going to show you in the next image, the solution to this problem has already been learned. I think you've probably had many a presentation on it. But we wondered in almost, again, going back to a tree-like situation, if B cells are antibody producers and T cells are T cell antigen receptors, if they relate to one another through a common precursor, and in turn there is a precursor to this lineage that also mediates a type of innate immunity called an NK cell, we wondered whether or not the variable region, the important working arm of the antibody molecule, may have been preserved in some form or another, maybe not rearranging, may be functioning on the surface of natural killer cells, which are very important in terms of recognizing viral invasion, tumor cells, cells that have been stressed through any number of different physiologic challenges. And that kind of reasoning took us in a very different direction. Now, while we were working on it, as just the last of the introductory slides, I guess there's one more later, that you need to understand that in man, one way that NK cell immunity is mediated is basically through a system in which immunoglobulin type, but not of the type that mediates, that affects adaptive immunity, immunoglobulin folded domains interact with another type of molecule called HLA that is expressed on the surface of normal cells, and it's highly, highly polymorphic. And there is basically, in a normal stasis, there is an inhibition of this NK cell from attacking that cell. But if a viral invasion takes place and we have a suppression of the expression of this particular marker, we have an activation taking place, and these immunoglobulin type molecules, completely unrelated to the effector molecules of antibodies and T cell receptor diversity type effectors, completely unrelated, will attack the cell. So that's the way it turned out in man. In mouse, it's effected, same process, same slide. You can now substitute in a different type of receptor molecule doing the same thing. We wondered many years ago, and as I said before this was known, if perhaps there was some relationship between that variable region system that proved so powerful in generating our immune diversity and in generation of NK type responsiveness. And this is the first of the animal models that I'm going to talk about in a little bit of detail. And this is a pufferfish. And a pufferfish's angle in the world of genomics is it has a very compacted genome. For some reason, they have lost very large numbers of introns, and they have lost very large, long lengths of intergenic space. And a pufferfish then provides, before the era of modern genomics, gave us a compact genome because of the nature of the way we were looking for genes, because its genome was small, and because introns create lots and lots of PCR artifacts. If you don't have very many introns, or shorter or fewer introns, then, or shorter intergenic spaces, you really should not have as many artifacts occurring. And we used a method that we developed called short primer PCR and turned up a very interesting molecule. A true V region with a true J region that was basically stuck into a mode not unlike what we later learned several years later are the human NK receptors. So it's almost as if it's a chimera between a molecule that looks like this and a T cell receptor, except of course these molecules don't really rearrange. And we learned later, much later on, that there are many, many, many of these genes. And in fact, we learned that there are 26 of them, of which 2 are pseudogenes. And they live in 12 families. And the families they live in look just like the types of families that T cell receptors and antibodies are distributed into. So they were a fascinating group of molecules that have been of great interest to us over the years. And we moved right away. Pufferfish is probably the world's worst animal model. One of the worst. I'm actually going to tell you about the second worst in a little while. The pufferfish basically eat one another, and they're toxic. Of course, pufferfish is not the same one, but it's fugu, which is the very deadly tetrodotoxin issue where you have to be super careful consuming these animals. And we turned for various strategic advantages to zebrafish to further study these molecules, and eventually to what's actually a better model than zebrafish, Medaka in many ways, a relative of sorts. And we now, when you can really look at a genome, once a genome is available and you know how to look at the genome, you learn very quickly that there are many, many genes. In fact, here we have our system. These again are our NITR molecules now being spread out in a manner to create 24 different families. And the diversity, in a Darwinian sense, the diversity of form is absolutely remarkable for people interested in gene divergence within multi-gene families. As you go across this group, we have inhibitory molecules, we have activating molecules, we have molecules with J regions, we have molecules with transmembranes, and some that don't have transmembrane regions. Every possible combination, plus again, throughout the 24 variable regions, I'll show you an image in just a second, all of this massive divergence of sequence. So this is a system that has undergone enormous divergence, but what struck us as being so peculiar is shown in this tree. Again, not quite like, probably better drawn than Darwin's, but nowhere near as clever. The distributions are that all of the zebrafish genes are grouping off by themselves in one way, and you can see them in green, and you can see the dramatic difference when you look at all the Medaka genes. So if you take 2 relatively closely related species, you see these enormous expansions of multi-gene families taking place, no doubt for adaptive reasons, within species that are relatively closely related to each other. I don't have the image with me, but if we put the third species that's informative onto this image, and by that third species, I mean the pufferfish, you would see once again a splaying out of completely different sequences with no relationship. So the same type of molecule is forming in each of these, and all of this divergence. This phenomenon is also true of immunoglobulins and T-cell receptors. It isn't as dramatic in terms of form, but in terms of sequences. And these are these huge expansions that take place in multi-gene families over relatively short periods of phylogenetic time. And the NITRs, I should tell you, are solely restricted to the bony fish, and that's an example of two things. That's a very large bony fish that I actually caught in a place that Darwin visited, and that's Patagonia in the background, a very, very unusual place. And when you read of Darwin in Patagonia, you can see not only the brilliance of observing how uniquely adapted species are to this very, very peculiar landscape, but then you read things like, well, I don't understand why these bones are found up in these mountains, and these mountains are rising from the sea. It all must have happened in the last several hundred years. It's interesting to historically read this absolute brilliance, and then Darwin being caught in these obviously monumental changes that are taking place in geography and not being able to reconcile it with his own beliefs. But the point of the image is that the NITR situation is truly one of that's restricted to the bony fish. We don't see it any further, and we know a little bit more about them. So the question you ask is, well, what are those molecules you've been looking at besides their interesting gene divergence? And this is, again, a ribbon molecule of a crystallized dimer that we described very recently, and it takes on the shape of an antibody molecule to a certain degree. And we know from very elaborate mutagenesis studies that NITR11, which is the 11th one in this particular system, is very effective at binding a target B cell. It has an allogeneic recognition, whereas a closely related structural variant of it called NITR10 really doesn't bind cells very well. And we know that we can mutagenize away that reactivity of NITR11 by changing a single asparagine to an aspartic acid. But more importantly, we can take a non-reactive molecule and simply mutagenize an aspartic acid to an asparagine and create a gain of function mutant. So the specificity, the NITRs obviously function in a manner that is very similar to that which you'd expect for a NK receptor, and that is the recognition of some allogeneic determinant. We, as yet, have not been able to pinpoint it to the level we would like. The mechanism of interaction, as I said, is a single amino acid difference. It involves a key region of the molecule that in the native form shown here is shifted with the introduction of an aspartic acid and a negative charge. And here we see our gain of function mutant. This is an ordinarily non-reactive molecule that has been created into a reactive form, basically proving the specificity end of it. And the picture is a little bit crazy. It was published this month, and those are the key species, which is another bony fish shown here. And the point of the title is, we didn't write that allo recognition by a primitive germline encoded antigen receptor. That's not quite true. It's probably not primitive. It's as modern as anything else, but it is very much like an antigen receptor, and it's very curious to us that what appears to be a function that resembles that which is effected by NK molecules has now come to be effected in by a completely different molecule if you look at a fish, if you look at a mouse, and you look at man. Huge, huge differences over very short time periods. Well, this is a strange view of deuterostome phylogeny. What it really is, it's a view from models that we have around to make conclusions from. It's really not at all correct in a phylogenetic. It's correct in a phylogenetic sense, but it's very restricted. There are many more species that factor in here. And the reason I want to just bring this up for a minute is here's our mouse human reference point. We've already dismissed the lamprey as being something that's completely derived in many ways, not just their antibody responses. It's a very different type of model. I want to focus your attention on Amphioxus, and Amphioxus is one of our key models that I'm going to end with today, and on the sea urchin. And Abe has already mentioned the sea urchin genome, and there also is an Amphioxus genome that we were involved with. We were involved with both of these genomes. And the last of the slides, and I think this one again for the immunologists, just bear with me for a split second, for the non-immunologists, this is probably the best known group of all of the innate receptors. These are the toll receptors. They do many different things, but they tend to have a very interesting feature that their reactivity is for relatively broad groups of molecules. Certain kinds of determinants on gram-positive bacteria, certain types of determinants on gram-negative bacteria, double-strand RNA for the endosomal molecules, single-strand RNA, etc. So we're not talking about 10 to the 14th specificities here that are uniquely specific, and you have experts here in the audience today who can tell you the complexities of understanding epitopes and which are the sort of the microscopic version of the determinant. These are much more generalized molecules that are factored very, very heavily in many aspects of our immune system. That is the purple sea urchin. The purple sea urchin got to be a genome project at first, not because of its immunology, but because of the fact that it is a very, very powerful developmental model. And like most genome projects, they get themselves onto the cover of certain types of journals. There's sort of an obligation by either Science or Nature to describe each one of these. I often wonder when that's going to run out, but there's just so much effort that go into these things. And every one of these species, when you look at these big phylogenetic differences, has something unique to offer. And there were many, many things to offer, but one that has caught the public attention that is very different has to do with these toll receptors. And in Strongylosis introitus, the sea urchin, we have an enormous amplification in the number of receptors. Now, there just aren't a lot of them in 214 versus your usual 10. It's not that you have a lot of these receptors, but they're diversified in terms of sequence in a very interesting way. They're not antibody molecules. There aren't 10 to the 14th, but they're not simple copies of one another either. There are definite patterns that are set up. There are differences. There are short multi-gene expansions, and there are basically translocations of single receptors to other parts of the genome. Some of them look a little bit like mammalian receptors. Many of them do not. They look like what you'd expect. And their key domain, the tier domain, is the common feature of a very large number of them. Roughly 70% of the genes are probably active. The same pattern shown here for toll receptors is exactly what you see in Amphioxus. So we have these two big sides of the deuterostome split, and these two groups of animals share something very unique and distinctive in nature. And that is expansion of different types of innate receptors. It's basically the same story. Amphioxus's expansion is probably only 80% of what it is in the sea urchin. Now, I said that pufferfish were a bad experimental model. Amphioxus are even worse. They live in Tampa Bay. It's the highest density in the world. They constitute 60% of or 70% of the biomass, and we have absolutely no idea how they breed, because the males and females are basically out of sync with one another. That's Amphioxus DNA, if you're wondering what just happened there on the screen. And the animal model is miserable. They can't really be kept in the laboratory for any great time, but they are a terrific genome model by any standards. And I think I have 5 features listed for them. First, their anatomy and development are very similar. They have many similarities with vertebrates and many similarities with other invertebrate forms, in this case, tunicates, who are a type of eukaryote. They are probably as good a source as any, and this is why we look at them, for what the ancient chordate genome looks like. The most interesting feature to me is they may have unique domain combinations. They will hook up a toll receptor to some other domain. Other domains that are involved in apoptosis appear in combinations that are completely unique to Amphioxus. And that phenomenon is just ripe throughout the species. As you look across the genome, there are all of these peculiar domain combinations, a very Darwinian idea of taking pre-existing information and just recombining it in a different way to make a slightly different organism. It is probably the most basal and reflective of the chordate lineages, and it also happens to have the highest level of allelic variation known to date. 6.8% across the genome is absolutely huge. It actually is greatly simplified, although your instincts are they would confound doing a genome sequence. It actually makes it easier in many ways. I can explain that. We developed a cloning technology called AmpTrap a number of years ago, and went in and found a molecule that had a variable region looking on a sequence level in terms of its core residues, not unlike that what you see in an immunoglobulin. I mean, you can never be really 100% sure, but it had the right length, it had some disulfides in the right place, and it sort of looked like a variable region. And there were actually two variable regions in the molecule. We call them V1 and V2. And then we have a chimera. We have a unique tail called a chitin binding domain. Now, chitin is the most ubiquitous bioorganic compound in the marine environment. It's polymerized N-acetylglucosamine. And maybe we'll come back to that in a minute. So we have a molecule that's sort of half something that looks like an immunoglobulin and doesn't rearrange, by the way. And then we have a chitin binding domain. And we knew then that by in situ hybridization that essentially all of the expression of the molecule seemed to be, of the gene, seemed to be localized along the intestine. There's an epithelial cell that certainly doesn't appear to be a structural cell that is interwoven into the intestinal architecture that appears to be the site of rather mammoth expression of VCBPs, and here's at a higher magnification. There, of course, is a control. We finally were able to make good sagittal sections, and it turns out that the pharyngeal area has a very focal pocket of an extremely high level of expression that clearly is just missed with the other sectioning technology, the other sectioning approach, I should say. And there are single round cells, very promising looking, that also express this, and those cells also engulf ink particles. So they're of interest to us, and I really won't talk about them too much. The main interest in these molecules came from observing these gene sequences you stack one on top of another. And what you see as we change the colors going down an image, a slide such as this or an image such as this, is that as you get to certain points, you have many, many different amino acid possibilities. And there, generally speaking, are a couple of regions of the molecule that really stand out. There's just a lot of diversity. And it spurred us to look in a little more detail as you went from one animal to another. So here we have 13 animals we're looking at. And the 13th animal is in the form of a library, a back library. It's actually the genome reference animal. And as you begin to look at the first animal, you're going to find anywhere from 5 to sometimes 8 or 9 genes. And it turns out they're copy number variants. We learned that last year. And the next thing you notice as you look at the sequences is animal number 2 has one thing in common, some things that are different. Animal number 3 has 3 things in common, 2 that are different. And if you start following this down, you begin to realize that every time we do another animal, there are new sequences that are appearing. Now, not that we're actually studying this, but it turns out that we have no less than 70 such situations that have arisen. And if I could create an image, which I can't, that would take you down 70, you will find that every animal in the population has at least one unique sequence in the variable region of the molecule. Well, when these things were first described, they were just perfect for speculation, and they got to be antibody molecules. They're not antibody molecules. They probably have very little to do with antibody molecules, but I always show this because it's interesting how people perceive things for basically the readership, and then you begin to wonder, well, if they did with something that you really know well, what else that you're reading might not be possibly so correct either. They had a major problem with them, however, that if you look at the germline of an antibody molecule, shown here, there are two regions of fairly significant variation that's germline in nature. And then I told you, remember how through this enzymatic process and a polymerase action, we have the introduction of basically random genetic information, creating enormous diversity in immunoglobulins and T-cell receptors. Well, the problem is that the variation in these molecules is really largely centered up on one end of the molecule into a lesser degree at an equivalent point in the second. This is one immunoglobulin domain. We could draw another immunoglobulin domain that has the same effect in it. And we turned our attention to protein crystallography of these molecules, and we were very, very new to this, and we were very, very lucky because we were able to achieve good quality crystals that had very high diffraction characteristics. And with a group, which I'll acknowledge later on, we were able to show that the manner in which this variable region folded really was somewhat similar to that in which T-cell antigen receptor folded, with the exception of one of the complementarity determining regions. But that when you examine these molecules, they really weren't antibody molecules as we knew them, because the antibody equivalent binding surface, the so-called complementarity determining regions, that analogous stretch is facing this way, and it's 180 degrees. Now, in an immunoglobular T-cell receptor, that CDR regions would be up on top of one another. So we have a situation where it really would be about as opposed as someone could imagine. However, if you then chart it on the molecule where the variation occurs, you can see along this face in gold that the concentration of the variation is actually creating itself on a different surface of the molecule. And this particularly, we're going to talk about that. So we have a molecule. The question I might as well talk about right now: what does the molecule bind? And therein lies a problem, because despite the fact that we know lots about its immunology and much more than I'm not showing you, for instance, it's very much upregulated on certain types of antigen stimulation. Cells that engulf things also express this at relatively high levels. I cannot tell you what the specific binding is, but we have some metagenomic approaches that are really working out where we're able to look at organisms that actually live in the environment of these animals, and that is a very promising approach. So back to some classic Darwin, and with a redlining is we have natural selection, and a very, very, I think it's the first time it occurs in the book. The preservation of all these favorable variations, the rejected injurious variations, will be called natural selection. And it led us to speculate many years ago, and it remains our working hypothesis, that in Amphioxus, what is going on is that because of the size of the population, which is estimated at some 1.2 trillion animals that live along a 484 mile coastline, and this is just one area in the world where they're fine, that we feel that if these are indeed immune effector molecules, which we sort of think they are, more or less, we have a unique survival advantage imparted to certain members of the population. Not really the clonal selection, but much more in a Darwinian fashion, an individual selective process. And the issue of diversity at this level, and I really can't do justice to it, fascinated us, and as the genome got resolved, we suddenly realized that the most variable of the genes fall in a group which are called the VCP25 genes, VCP25 group, and it consists of 3 genes in most animals, although there are copy number variants, and those genes are known as A, B, and C. And if you look at A, B, and C in a little more detail, and these are just hydrophilicity plots of these animals, and we certainly can look at stacked up sequences, most of the variation resides in the A molecules. And these are just some of the tracings out of these molecules. It's just a different way of showing what kinds of amino acids are present rather than showing tedious amino acid sequences. But we have one of the types of molecules, the A region, the so-called A genes are highly variable. The C genes all basically look alike. The B genes fall into 2 families. So we have a gene family, and it turns out that one of these genes is being is for some reason undergoing a lot of genetic change. Now, one last level of complexity is something about that change that is very unique, because we know that some haplotypes of the animal have enormously high, and these plots are all based on the length of scaffold regions from genomics, so you can't really look at it the way you're used to using data. Your best bet is to look at it around a narrow sector. It turns out that some of these haplotypes have enormously high levels of inverted repeat DNA around them. And one of them in particular is the richest region in the entire genome of inverted repetitive DNA, which very well could be a factor in promoting genetic change in these animals. And we happen to know that that very well could be the case, because when you look at isolated sperm from these animals, you find rather extreme rates of meiotic recombination taking place. So what we think is that whatever these molecules do, they're out there. The population is largely stable, but there are certain genes in the population, certain haplotypes, that are far more prone to others than developing rapid genetic changes, and our working hypothesis is that it very well may be related to inverted repeat DNA, and that actually is known to influence such processes. So we know from Darwin, in this is the last of the Darwin statements, is there is a very, very elaborate statement on long geologic ages and seeing differences from what they previously were on a rather protracted scale in the classic Darwinian understanding of what was available then. Now, of course, in the case of the finches, it's probably a lot faster than Darwin thought it was at the time, because he simply made some observations. But had he actually stayed there and had the knowledge to conduct the types of ecology experiments that people do with them, and of course with no understanding whatsoever of genes, I think the basis for that is far faster than he would have thought. So the differences in our immune system, which are now depicted once again, where now you sort of have my permission to look at the whole thing, are that the changes over relatively brief periods in metazoan evolution are really dramatic. And we've dwelled on the case shown here of man and mouse, contrasting it with a completely different mechanism, same molecules, different mechanism, same molecules, same mechanism, different organization, complete switch in operational mode. We go down to Amphioxus, or we go over to Amphioxus would be more accurate. Amphioxus showing possibly the utilization of alternative V regions. These massive expansions taking place in innate immunity in animals such as sea urchin and in Amphioxus. And now you go to an animal, a mollusk, and in a mollusk there's a molecular family called a FREP family that is specifically reacted with the parasite coats in these animals, and these particular molecules have extraordinarily high rates of somatic mutation, an effect that you really see in the vertebrates, actually in this vertebrate, in that vertebrate, not so much in that one. And finally, when we go to Drosophila at the far left, certainly there's innate immunity. The toll receptors were certainly seen there first. but one of the chief players turns out to be differential RNA processing of a molecular family, DSCAM, that's also actually involved in neuronal patterning. So what has happened through this enormous passage of evolutionary time is that all of these different mechanisms have arisen to confront the pathogen. And the driving force here isn't going to be whether or not a highly adapted land mammal develops a bigger foot for better swimming to move from one region to another, or a slight shift in oxygen tension is going to improve. The difference here is that these are life and death situations that take place in this constant struggle between pathogen and host. And it really comes as no surprise at the end that all of this variation that we're achieving along this side in the host immune responses, somatic recombination, hypermutation, all of these generalized variations that are taking place, as well as epigenetic processes, are really taking place in response to pathogens that, even without invoking many of their other clever strategies, have their own forms of variation, recombination, and etc. Strikingly similar to that, and all being played out through these host immune type cells. So at the end of the day, what would Darwin think? Well, he probably would not have had much of an appreciation for adaptive immunity. It certainly took us long enough to figure it out ourselves. I think, on the other hand, the innate system would have been far, far easier to grasp in the typical logic that was applied in the other systems that he looked at. Well, without taking any more of your time, and certainly thanking the people not only in our own group, but in the many, many collaborators, largely work with John Cannon and Larry DeShaw you've heard about today, and some of the other collaborators. Jonathan Rast is our sea urchin collaborator. Andrew David Ostrov's lab are the crystallographers. Jose Hernandez, Chris Amamiya on the back libraries, which figured so heavily in all genomics, and especially ours. Many of the sequence analyses were done by Tatsuya Oda and Lee Rowan at Lee Hood's Institute, and Gustavo Glussman on many of these assemblies, and then of course Adam and Ching on the molecules which I really didn't do justice to, and that are these peculiar domain combinations, an adaptive feature of Amphioxus. Thank you for your time. Yes.
So, what would the politicians argue in terms of putting resources into studying man, mouse, current immunology versus what we learned from evolutionary?
Well, I think it's not the first time I've been asked that question. It might be like the 150th. But to answer your question, the value of these systems, you could, I'm sorry?
Repeat the question.
The question basically, and I'm going to just paraphrase it, is what would politicians say of funding this type of research or devoting resources to this type of research versus research on more of a, let's say, human and maybe mouse mode? Is that a fair?
Well, this is part of the question.
Yeah.
Which drives which? Does the knowledge of the mouse-human lead to the knowledge of the older organisms, or does the older lead to
Okay, that's a very different question. I think prior to genomics, it may have been very much predicating work on what you knew about man and mouse. But with genomics, what comes out of it, for instance, this haplotype stability issue, that was not a predicate that you would go into an experiment with. That came out of it, and now actually has led us back to a human predicate. So it works in both directions. One of the problems in this type of work is really, what are you seeing that's different? And And I think what comes out of it is when you look at a model like that DSCAM thing that I treated so briefly, here you've got a molecule that processes out into some 30,000 different isoforms. The next question you have to ask yourself is, gee, that's going on in Drosophila, but I wonder if there's other massively diversifying systems that also might be creating molecules that we're not predicating our experiments on. So it's about as high risk a type of research as you could do, would be my answer. But I think at the end of the day, gene conversion, clearly an important genetic mechanism. Gene conversion, in the modern sense, was discovered by someone in France looking at an avian model. And now it's factored in very heavily. So it's pure basic science. That was a good question, though, the part that I didn't quite catch the second part. Cliff.
So if you see the major expansion of gene families as like folate receptors in the sea urchin, I'm naively thinking about this. It's not an evolutionary biologist or geneticist. It seems to me that one possibility is that there's a functional expansion that is related to kind of called the original function of those genes and proteins. The other possibility is that they're really being used for other things. And one that comes, could you answer that in general and also in a specific example of that related to the TLRs would be, for example, the use of the tear domains in receptors like the IL-1 receptor, which is really a different, has a different function than the folate receptors. Is that happening in the
You have no, we don't know that. Where they very well could be functioning, although they really look like they do in mammals, is in development. They may be playing an important role in development, but the reason we don't think that's a very strong hypothesis is that sea urchin is the very perfect model for looking at that. And when morpholinos directed to the toll receptors in a very specific way, and one of the beauties that we have in these systems are the use of these inhibitors of translation initiation, they really don't impact at all. And there's all kinds of controls, of course. There's morpholinos that will completely change, can alter sea urchin development regularly. So it might be wishful thinking, but it's not just us, it's others have this same feeling that they just kind of look immune-like, and there's much to be learned. I mean, these were just discovered a little while ago. Neil?
There is evidence for toll-like receptors, or these putative toll-like receptors functioning in development. The bobtail squid, where it has an organ called the light organ, which is just to illuminate the bottom of the sea so it doesn't get seen by casting a shadow.
Right.
And it turns out that there's an interaction between resident bacteria and some receptor, which may turn out to be a toll-like receptor, that is essential to the development of the light organ. So it's not confirmed yet.
Yeah.
It's a toll-like receptor, but it seems like
That's
the LPS, which is eliciting the interaction.
good point. Anything's possible.
I'm going to do something silly. I'm going to sing a fragment of a song that my mother taught me. And see whether you, Gary, or anybody else knows it.
Okay.
It's, goodbye to fins and gill slits. Welcome lungs and hair. It's a long, long way from Amphioxus, but we all came from there.
That's way better than the seminar.
Your mother.
Great song. Yes.
Are monocellular organisms capable of immune response?
Ruslan Mezatov thinks they are. He's one of the leading figures in toll receptor. I can't answer that. I don't. The question's been raised. I don't know the answer. I mean, I think it all depends on how we want to define immunity.